So it's my pleasure to introduce Rex Jung. Do you go by Jung or Jung? Jung. Assistant professor of neurosurgery at the University of New Mexico and a research scientist at the Mind Research Network, an amazing, fabulous place in Albuquerque, where he's also a practicing clinical neuropsychologist. His research is dedicated to relating behavioral measures, including intelligence, personality, and creativity, to brain function and structure in healthy neurologic and psychiatric patients. And again, the MRN, or the Mind Research Network, has become one of the nexuses of both analytical creativity. Certainly it's one of the central places for independent component analyses, but also for this idea of creating large networks of scientists who can aggregate their data, their methods, and have a database that's also quite transportable, et cetera. So welcome.
Thank you very much. Thank you very much. So with a name like Rex, I guess it was inevitable that I would end up in the western part of the United States. So I'm from Albuquerque, New Mexico. With a name like Jung, it was inevitable that I would become a psychologist, or a psychiatrist. And I have no idea why I'm studying creativity, but here we are. To kind of riff off the last talk, perhaps I do have an idea. Before I entered graduate school at University of New Mexico, I was working in the laboratory of personality and cognition with McCrae and Costa and Leonard Giambra, who none of you have heard about, but he was doing research in task-unrelated thoughts. And we were doing research that was sitting older subjects in front of a screen and just boring them to death and measuring when their minds wandered. So perhaps this is coming full circle. We saw in the last graphic this measure, task-unrelated thoughts, or TUTs, are part of this default mode network, this very early conceptualization of mind wandering and task-unrelated thought. So perhaps we've come full circle. So the organization of my talk is here. I'm going to go over what is creativity. We haven't really talked a lot about creativity yet, and I'm going to wander a little bit, so I'll raise my voice when I do. It's the production of something novel and useful. This is our best definition of creativity, and it comes from the 1950s. From Stein, and this concept, this interaction, this interplay of novelty and usefulness is important. This dynamic interplay of novelty and usefulness is particularly important in the study and the conceptualization of creativity. Can we see it in the brain? I think we can, but the struggle is to transcend this concept of neophrenology or neurophrenology. And while networks are important, we shouldn't devolve into this neurophrenology that we neuroscientists have a tendency to get into. And so where is it? The answer is we don't really know where creativity is in the brain. It likely takes all of your brain to be creative. It's certainly not in the right hemisphere, as you've heard, but networks certainly help. Networks help us understand where creativity is, how it's manifested in the brain, and how to move forward. It gives us a good first approximation about how we can study creativity from a neuroscience perspective. So first, what is creativity? We can look back to Henri Poincaré, a brilliant mathematician and physicist who looked into his own mind and tried to understand how his creativity was manifested. And the thing that I want to instill into you is that creativity is not one thing, but it's many things. It's many cognitive processes. So when we talk about creativity from a psychological perspective, it's not going to be this one magical thing that we arrive at. And so Poincaré talked about these different stages, these different processes of creativity, these different cognitive constructs that we might want to fractionate creativity into. The first one, preparation. You want to prepare. The creative person has to put some ideas into his or her brain in order to be creative with. You have to have the raw materials. If you're an artist, you have to understand something about color and hue and tone and perspective. If you're a scientist, you have to understand the scientific method. If you're a neuroscientist, you have to understand something about the brain. You have to prepare and put some ideas in your brain to be creative with. This 10-year rule is probably important. It takes time. Malcolm Gladwell popularized this, but it came earlier. Hayes in 1989, Cognitive Processes and Creativity. It came earlier, this concept, this 10-year or 10,000-hour rule, that it takes time. It takes long periods of time to prepare to be creative. How are we going to get at that from a neuroscience perspective? How are we going to understand that? This is a very complex problem that we're trying to solve. Incubation is probably what we're talking about in this default mode network. This is the long walk of Beethoven, this ruminative process that people go through when they're thinking about a problem that they want to solve, this internal problem solving that you've heard so much about in these fine talks today, this internal cognitive process, perhaps unconscious, where people are mulling over a problem, this cauldron of ideas that are spinning around and forming and combining in novel ways. Kekulé talked about awakening from a dream and thinking about a snake swallowing his own tail, and discovered the benzene ring. I would even offer that Hemingway had some ideas about how to induce this period of downregulation of the frontal lobes by drinking. He talked about you want to write drunk and edit sober. But this idea that you're downregulating the normal control mechanisms of the frontal lobes that constrain your ideas, this idea generation part, this incubation is incredibly important and probably has a lot to do with the default mode network. Illumination. There are researchers that are studying that aha moment. Archimedes had this eureka moment where he was in the bath and he discovered that he could measure the amount of gold in a crown by submerging it in the bathtub as he was himself submerged in a bathtub. This moving from unconscious to conscious processes is an important part of creativity. This illumination, it's not critical that you have an illuminative moment, a eureka moment to be creative, but some people do, and we need to understand that in terms of this creative process. And then verification. You have to check to see if your idea truly is useful out in the world. If it is going to work, is it going to be adaptive? Are people going to accept it? Oftentimes, as scientists, we put out research ideas, and I don't know about the rest of you, but all of my research papers have been rejected the first time around, my creative product, and you have to go back and keep telling them that this is a good idea, this is an idea worthy of being published in the scientific literature. You have to persist, you have to have grit, you have to have determination. These are personality variables that we have to understand in order to understand creativity. So that creativity is not one thing. Creativity is many things that we have to understand from a psychological perspective, from a neuroscientific perspective. And this is a very complex problem that we have to solve. Now, from a cognitive neuroscience perspective, Arne Dietrich has looked at this another way, that there are also knowledge domains that we have to understand and processing modes that we might want to take a look at when we're looking at creativity. In the knowledge domain, you have cognitive types of creative capacities that people are pursuing, and you have more emotional types of creative things that people are producing. In the processing mode, certainly there's deliberate creativity. The scientist is working on that paper. The artist is working on that drawing or that painting over long periods of time. And there's more spontaneous aspects of creativity that are important to understand. So to populate this, Marie Curie perhaps is a good example of that cognitive, deliberate person, the scientist who is toiling away. Their creative domain, if you will, is that cognitive world as opposed to the emotional. Now, this doesn't mean that they don't delve into emotions at all, that it's purely cognitive. You can go back and forth between these boxes, but it's primarily in the cognitive domain that she works. Pablo Picasso, artists more working in the emotional knowledge domain, where they're either expressing their own emotion or they're trying to elicit emotions from other people. Miles Davis. So, improvisational musicians are incredibly important to understanding this creative process. And there's been good work by Lim at Johns Hopkins looking at individuals doing improvisational activities in the fMRI scanner. And what you see is really interesting, just to diverge quickly, is this flipping back and forth between the cognitive control network and the default mode network. That when people are improvising in the scanner, that they're actually down-regulating the cognitive control network, and the default mode network is coming alive when they're improvising. This is very interesting, important research in the creativity literature. Finally, the cognitive spontaneous. I mean, Isaac Newton, when he's sitting under the apple tree and the apple falls, and he's been working on this problem for a long time, and he thinks about gravity, this type of aha moment that scientists in the more cognitive domain have. This is important to understand as well when you're looking at creative cognition. Again, it's not just one aspect of creativity that you're looking at, it's many. Cognitive and emotional knowledge domains, deliberate and spontaneous processing modes. I hope to instill in you this necessary constraint that we have in the literature and in the field, that we're looking at a very complex problem, and we might want to fractionate it into different pieces. We've really focused on certain aspects of creative cognition at the expense of others. Perhaps that's why we're seeing the default mode network come up so often. But if we start looking at these others, we might see the default mode network be an important component of creative cognition across the board. And you might see the fractionation, as some of the speakers have said, including the last speaker, of the precuneus, for example, and the more dorsal aspect of the precuneus being more important to cognitive deliberate, and the more ventral aspects, if you will, being spontaneous deliberate. You might be able to fractionate it further and see which particular aspects of the precuneus or particular regions of the frontal lobe within the default mode network are associated with manifestations of creativity. Plus, we need to think about creativity as an evolutionary thing, as a reasoning capacity. How did creativity emerge? I mean, this dolphin is playing with bubble rings, which I find fascinating. Why is it doing this? This is a creation of the dolphin, and it's not just these dolphins, but beluga whales can do this. Different types of dolphins can do this. If another dolphin comes close while it's playing with its bubble ring, it will pop it. You're not playing with my bubble ring. This is a fascinating behavior. Is it creative? Is it novel? Is it useful? What's going on with this particular dolphin? What is it thinking while it's doing this? Is this a type of reasoning capacity that other animals share? And if we're going to look at creativity in humans, we should link it, as Dr. Buckner did initially, across species and across evolutionary time. There should be some reason that creativity emerged in the first place as a capacity that has remained. I don't think it's just a spandrel that has resided within us through some accident of evolutionary history. Another example from dolphins. This particular dolphin, Tursiops truncatus, that's found in Australia, I believe, was discovered to use these basket sponges to help itself forage for fish in sandy beds. This is tool use straight up. It has a very sensitive beak, and it's rubbing its beak on the bottom of these sandy beds to flush fish out from the beds, and then it can drop the sponge, eat the fish. This is tool use. How is this different from the hand axes that we saw in Dr. Buckner's talk earlier? This is an evolutionary constrained behavior. This is creative. The dolphins are doing it. We need to understand why. So I'm starting to look at creativity as from an evolutionary perspective as well. And I think, even though psychologists go back and forth about loving or hating Cosmides and Toomey, there is reason to look at different fields and different perspectives when we're talking about creativity and something as complex as creativity. And they say elsewhere, we have written at length about the trade-offs between problem-solving power and specialization. General purpose problem-solving architectures are very weak but broad in application. whereas special purpose problem-solving designs are very efficient and inferentially powerful, but limited in their domain of application. So they hypothesized that there's two reasoning types of system. Because of all the problems that need to be solved out in the world, you might need these two types of reasoning system. One that is a rule-based cognitive system that is very rapid and accurate for reasoning out problems that you encounter frequently in the environment. This is kind of like intelligence or deductive reasoning type of ability, and one that is situation-specific improvisation. That's novel and useful reasoning, something like creativity. So from this, you get things like induction, abduction, poetry, art, music. This is a possible mechanism by which you can have creativity emerge without it being purely a spandrel. You have different reasoning needs that are created by the environment. One that is very general in purpose and one that is specific. And I think this is something worth pursuing. So, a little talk about statistics. This is not going to be as pleasant as the first part of the talk. And you've all been exposed to statistics, as I have repeatedly, because it didn't take the first time. But we have issues of reliability and validity. And reliability, and we're going to play some target practice, and we're going to talk about triangulation in neuroscience. This isn't just a relationship issue that Bill Clinton is guilty of. This is stuff that we would like to talk about that might be useful to the cognitive neurosciences. So reliability is measuring something consistently, and validity is measuring what you intend to measure. Now, if we were to do some target practice and look at something that is valid but not reliable, it might look something like this. We're aiming for a target, we're hitting the target, but we're not hitting it in a very consistent way. It's kind of all over the place. Our aim is kind of a scattershot approach to this. In contrast, if we have a highly reliable measure, but it's not valid, it's very tightly bunched together, but it's not even hitting the target. We can measure something in a very consistent way, but it's totally missed the point. It's not hitting what we intend to measure. What we're really looking for is a measure that is reliable and valid. And why am I walking you through this kind of dull statistics review? When we're looking at something as complex as creativity, we need to have reliable and valid measures. And frankly, most of our measures in creative cognition suck. They're pretty bad, and I'll show you that. I was horrified when I entered the field how bad the measures were from a psychometric perspective. And then also from a neuroimaging perspective, as I'll show you, there's some problems as well. Now, I don't want to have a fight with the FMRI folks. And although I think I could take maybe one of you, there is a problem with functional magnetic resonance imaging in that it has rather fair reliability. Not great, not poor, but rather fair reliability. When you put someone in the scanner and you ask them to do a task, and you take them out and you put them back again and you ask them to do the same task, you might get a different answer. It's about 0.5. So even Dr. Shalla said earlier today that we really don't even know the reliability of the default mode network. And I think that's a really important thing to understand. How reliable is this network that we're trying to get a handle on, and how can we hope to correlate that with creativity with other unreliable measures which are even lower in the creativity literature. So we need to try to use reliable and valid measures. Now this is why, and again, we wouldn't be here today talking about the default mode network without functional magnetic resonance imaging. This is a fantastic technique, an important technique, and a technique that has brought the field far forward in the last 20 years, farther than we otherwise would have been. So I'm not denigrating functional magnetic resonance imaging at all. But this is the target of the creativity literature so far in a review that we did in behavioral brain research in 2010. There were only 7 studies at the time, and it's a little hard to see. But you can see that we haven't really hit the target. There's 7 studies, and it's kind of a scattershot all over the place, including something very important going on in the cerebellum of the brain in this one study, which I don't really fully understand. How can you have all these activations in the cerebellum of the brain and it be related to creativity, and not in these other studies? But certainly if you look at these other studies, there's lots of medial frontal activation in the default mode network, probably in the inferior parietal cortex in the default mode network, and certainly in the temporal poles in the default mode network. So in the other studies, there is some correspondence, even in the fMRI literature, of creativity studies and the default mode, even in this early review that I didn't appreciate. So I'm proposing that we do some triangulation in neuroscience, that we try to use multimodal neuroimaging techniques to get at this very complex cognitive construct. One of the techniques that we use that I'm sure you haven't heard about is proton magnetic resonance spectroscopy. And this is where I cut my teeth when I entered graduate school at the University of New Mexico. I happened to fall in with spectroscopists. And what is this? Well, this is a way to measure biochemistry in the human brain using a standard MRI machine. And I fell in with Bill Brooks, an Australian, and the first thing that he told me to do when I suggested to do a study was, what's the reliability of your measure? Put someone in the scanner, scan them, take them out, put them in again, see what the reliability of your measure is. Even before I did an experimental study correlating NAA, N-acetyl aspartate, which I'll talk about in a second, with a behavioral measure. So proton spectroscopy allows you to look at a particular region in the brain, and way back in the day we could only take one voxel of tissue and we could interrogate it very well, maybe 10 minutes of time interrogating this big old chunk of brain deep in the white matter, and you get this characteristic spectrum. And water is the most predominant signal, but based on the number of hydrogen atoms, protons, that are in a particular molecule, you have different other metabolites that resonate slightly off frequency. We can magnify these, we can measure them, and you can get a chemical assay of certain chemicals in the human brain, which are interesting to us. In proton spectroscopy, now we've become more sophisticated where we can actually take a grid of different regions in the brain, and we can, although we can't get out to the edge of the brain, which is where all the action is, we can interrogate interior parts of the brain and get a grid, each one of these a different number, a different chemical signature, including the anterior cingulate gyrus, posterior cingulate, which will be of interest to people in the neurosciences. Now, NAA is one of the chemicals that we happen to be able to see. And it's interesting because it's found predominantly, almost exclusively in neurons. So it's a neuronal marker of neuronal viability, neuronal density. It's the second most abundant metabolite in the human brain after glutamate. It's turned over about 1.5 times a day. Its synthesis is directly coupled to glucose metabolism, and it may protect neurons from osmotic stress. So it's a really good metabolite that we study in disease, traumatic brain injury. I've studied lupus. You can look at tumors with NAA. But we were the first group to actually correlate it to normal human functioning, intelligence. I went to Bill Brooks and I said, I'd like to do a study where we correlate NAA. It's correlated with all these other neuropsychological measures in disease. Has anyone ever correlated it with normal brain function? Well, no. Why would you do that? Because we're cognitive neuroscientists. We study normal brain functioning. Oh, it's a big waste of time. Go do a reliability study. I did that, and then I correlated it with intelligence, and we found some interesting findings that you can read elsewhere. Structural magnetic resonance imaging also has high reliability, and we use a technique called free surfer. This allows us to take individual MRIs, which you've seen on most CSI programs, and convert them into renderings of brain, and this is a rendering of my brain to prove that I do have a brain in my head even today, and do these beautiful parcellations, if you will, of different regions in the brain, different gyri, different sulci. And we can, if I expand this out a little bit, you can see that we're able to look at and measure the white-gray matter difference. So we're actually able to see the cortical mantle, the thickness of the cerebral cortex. And we're able to, across the entire surface of the brain, correlate cortical thickness with some external measure, be it intelligence, creativity, personality variables, and we've done these various measures with free surfer and structural magnetic resonance imaging. Diffusion tensor imaging is the third major technique that we use, and this is an example of probabilistic tractography of white matter that is taken again from my brain. So I do have white matter as well, going through the corpus callosum, showing you the connectivity of different regions in the brain. We can articulate a seed, move it around, and show you the probabilistic connectivity of different regions of the brain, which is very beautiful. This is not a scientific technique, at least in our hands. We use a much more basic technique, which is tract-based spatial statistics, which allows us to look at the major white matter pathways in the brain that are connecting up these cortical regions and look at the connectivity of these different regions in the brain in normal human subjects. And again, almost all the research that we do is in college subjects, the lab rat of the modern neuroscientists. So we have ready access to college samples. We're not studying highly creative people, and we're not studying uncreative people. We're studying college students as they, and we're also studying the creative tasks outside of the scanner and correlating it with these measures inside the scanner. And fMRI is a valuable and useful technique, and we are getting a rest scan from these individuals as well. This we're able to subject to graph analysis, to independent component. Anyone who's within 100 miles of Vince Calhoun must do independent component analysis, so we're obviously doing independent component analysis on this data and subjecting the data to these broad networks and looking at the data in that way, this REST scan, which is not task dependent and very interesting. So, where is creativity in the brain? Again, we don't really know, but we have some first approximations about how creativity might be manifested in the brain. But first, it depends on what it is, and this is kind of the suckedness that I'd like to walk you through in terms of the measures that you haven't heard much about in our talks so far. And the measures are okay, but they're remnants from this notion of divergent thinking that's been around since J.P. Guilford's APA, American Psychological Association, presidential talk in the 1950s. And he really made a call in that talk to treat creativity as importantly as a psychometric measure as intelligence had previously been treated. And people really took that to heart, and he gave examples of how one might go about studying creativity as something different than intelligence, and divergent thinking was one of these measures that he talked about and helped create. So how does this work? Well, this is the classic matches test, and the matches works like this, where you have this set of matches. You are to remove one match to make 7 boxes. Who has an answer to that? That's a little hard. You can move match B, C, T, or U, and that will leave the 7 boxes. And you have different types of problems like that, where you're to remove 1 or 2 or several matches to make a certain number of boxes. So this is considered a creativity measure. It's not a great creativity measure, but it's considered to be a creativity measure. The classic measure of creativity is this: tell me as many different ways you can think of to use a brick. And so people say, well, to build a house, to throw at my brother as a mock coffin at a Barbie funeral. Well, that one's different. And you get these different responses, and the number of responses are important. The fluency of responses is an important measure. Fluency often leads to greater originality, as Dean Keith Simonton has done a lot of research showing that the number of ideas often leads to better quality of ideas. Frequency or different types of ideas, using a brick as a weapon or using it as a decorative item or using it to that extent is another measure. Originality is really where the meat of the matter is. How original is the idea? How novel and useful it is? And then elaboration is the last. Tell me as many different ways you can use a paperclip. Our measure of insight is pretty funny as well. You have these 3 words: lick, mine, and shaker. And you're to think of one word that binds them all together. Can you think of it?
Salt.
Yes, How did you do that? It just popped into your head in a moment of insight. And that's how this is considered to be a test of insight. It's not a process of deduction or process of elimination. It just pops into your head as this semantic thing that you know binds these words all together. This is our measure of insight. It's pretty sad, huh? So we also do things in the fMRI scanner. Tell me, think of as many novel uses for a pen as you, think of a new design for a pen. This is one of the classic examples, a new design for a pen. And they can run the scanner for 12 seconds, and here's another example of a pen, give you an example of a pen. Think of a novel, new design for this type of pen. And you can look at novelty and originality and what people are doing in the scanner this way. And then people just straight up measure the Rorschach ink blot. You can look at the ink blot and have people either give common responses. The psychoanalyst in the audience will know this Rorschach test, and I was trained on this as well. Common response to this would be it's a butterfly. This strange crowd. Or cherubs at the Trevi Fountain holding up a vase. So you can have very common responses, a bat, a butterfly, or more unique responses or original responses. And you can look at the difference between those types of responses to a Rorschach ink blot, a very non-defined type of stimulus in the scanner. So if we use the same measures, this is what we found. And this is kind of a brief summary of some seminal studies that we did a few years back, starting in 2009. Looking at spectroscopic measures, we studied about 60 subjects at this time, and we asked the question, where in this grid of biochemical measures, N-acetyl aspartate, this measure of neuronal integrity, where was this related to a composite measure of creativity? We put all our measures of creativity together, divergent thinking, and where were they related to each other? And it turns out it was related to a region in the anterior cingulate cortex, and it was related in an interesting way. The relationship was inverse in average intelligence individuals, and I won't get into the details of that now, but the relationship was inverse, higher creativity, lower NAA. We interpreted this as a disinhibitory relationship in a semantic access to semantic networks. Very interesting, this inverse relationship, higher creativity, lower neuronal integrity. We also asked a question, slightly larger number of subjects. We kept collecting data, maybe we're up to 67 or so now, and we published this in the Human Brain Mapping, where we asked the question, where was cortical thickness related to our measures of creativity? And we found a region in the frontal lobe where there was a statistically significant relationship, but the relationship again was inverse. Cortical thinning was related to higher creativity in our experimental sample. Now this is getting weird, because it's usually bigger, better, stronger, faster. Cortical thickness, better functioning. This is what we found in our intelligence studies, but creativity was telling us something very different. Finally, in the white matter findings, we asked the question, where in all of the white matter tracks was creativity related to integrity of the white matter? As you can guess by now, there is a region in the frontal lobe, the anterior thalamic radiation, where we found an inverse relationship between the integrity of the anterior thalamic radiation and our measures of creativity. By the way, there's a region in the right hemisphere where openness to new experiences was also inversely related to integrity of the white matter in the right hemisphere. Now this is getting really strange. So we wanted to look at the breadth of the structural neuroimaging literature. We'd already looked at the functional imaging literature and seen that it was kind of a scattershot all over the place, and we really didn't understand that. So we looked at the structural neuroimaging literature, and this includes lesion studies, and there's this very important series of studies by Bruce Miller out of UCLA, I believe, who's looking at patients with frontotemporal dementia and finding that patients with particularly deterioration of the left anterior temporal lobe have increased creative capacity, increased artistic ability in particular. Not all patients, but some patients, particular patients, have increased creative capacity. You have our studies, which I've summarized here. You have Shamed Sori, I believe, is how you pronounce that, did a lesion study in 2011 where they're looking at individuals with brain lesions, running these divergent thinking tasks by them, and finding that a particular lesion to the inferior parietal lobe, the default mode network, is associated with increased creative capacity. All the regions in blue, there was about 6 or 7 individual groups that we were able to summarize, and all the regions in blue are regions where lower measures of brain integrity, whether it was a lesion, lower cortical thickness, lower white matter density, are associated with increased creative capacity. And of course, there were a few regions in red that were associated with increased creative capacity as well. What does it all mean? In this review, we talked about, and this is why I call my talk networks of creative cognition, because I don't think it's one network, but I think because this problem is complex, we're going to need to talk about several networks. And this internal thought network that you've heard so much about, this general purpose problem-solving network, a situation-specific improvisation, this creativity type of network, induction, abduction, this mental simulation, the ability to try problems out, to try simulations out in your mind's eye before you put them out in the world, is probably constrained by the default mode network somehow. This is very important information, and it probably has something to do with preparation and incubation, as we talked about in terms of the different stages of creativity. The external thought network, this special purpose problem solving, is rule-based cognitive system, deductive problem solving, cognition in the external world, and is probably related to illumination and verification components of creative cognition. And I've also hypothesized, as someone mentioned earlier, that the salience network, or something like it, is probably involved in switching back and forth between the two. These improvisation studies tell us something very important about the networks do switch back and forth, but how? How do we find salience in the environment? How do we notice that something has changed in the environment so that we have to switch back and forth between this internal problem-solving mode and this external problem-solving mode? That's an important discovery to be made. Finally, the way forward. Okay, creativity is a complex cognitive construct. It will be important to fractionate this construct if we are to understand it. Novelty versus usefulness, unconscious versus conscious thought. What's going on in that unconscious process where ideas are just spinning around in this cauldron within the default mode network? Incubation versus verification, simulation versus action out in the environment. I think this is the default mode network versus the cognitive control network. We need to understand how these flip back and forth, how these relate to each other, as the earlier talks alluded to. We should use reliable, valid measures. Behavioral measures are way too focused on divergent thinking. Illumination measures are poor. Our measures of insight, as you saw, are particularly poor. I think improvisation holds great promise. Putting people in the scanner and having them improvise and looking at this process holds great promise in understanding about the creative process. And triangulation is key. Lesion studies, structural magnetic resonance imaging, DTI, spectroscopic imaging, FMRI, EEG, all of these should be used to understand this complex cognitive construct. And brain networks are certainly better than looking at bits of brains to try to understand creative cognition. I think that's allowed me to constrain the problem a bit better. Now, as opposed to doing these grand fishing expeditions, which I've done in the past across the entire brain, I can focus on the default mode network or the cognitive control network, have higher power to look for significant results in the future. So we're also using graph analysis. I have a very bright student, Sefer Ryman, who's we're collaborating with Martien Vanden Heuvel, who's a student, former student of Olaf Sporn's, and we're using this complex graph analysis to interrogate our data, particularly the diffusion tensor imaging data, not the functioning data, but as Dr. Shallis, I believe, said earlier, you can kind of feed any data in there, fMRI, structural data, diffusion data, and we're looking at the connectivity between these different hubs and finding some interesting results between the efficiency of the connections, the connectivity of the connections. We presented this at Society for Neuroscience last year, preparing this for for publication currently. This will get us in trouble because it appears there's a difference between males and females in terms of how they're using these different networks to come to the same level of creativity at the end of the day. Finally, I would ask you, those of you in the audience who are squeamish, to turn your eyes away because I'm going to show you, I work with neurosurgeons, So I have the opportunity to do awake craniotomies. And awake craniotomies allow you to actually tinker with the machine. And we do awake craniotomies to understand where in the brain the particular lesion is. This tumor, this is a patient that we did an awake craniotomy with. And we can map out, the different regions that are active in terms of language functioning, and the regions that the surgeons can take out along with the tumor. Because of where we often do these surgeries in the anterior Broca's area and the anterior temporal lobe and the inferior parietal lobe, these eloquent areas of the brain, I think this has enormous opportunity to do some functional neurosurgical studies with creative cognition, actually with design studies doing stimulation of different regions of the brain while someone is doing a divergent thinking task or an insight task or another task that has greater reliability while someone is actually in a surgical suite. I think this is very important research to do, and I thank you for your attention.
Thank you. Please.
We need it on?
Yeah.
Oh, check. Okay. Hey, great talk. I noticed it was a little short on EEG, and I'm wondering why you think that might be. For instance, the situation where you put the Lick Mine Shaker up there. I was thinking of a song that Mozart wrote. It's not well known. It's called Lick Mine, other word. But nobody really knows that that was Mozart, actually, but it was. So it was a different association that I had that she had.
Yeah.
It happened like this, though.
Sure.
And it happened just as quickly for her as it did for me.
Yeah.
With MRI, though, you're measuring over long periods of time, seconds, tens of seconds, right?
Yeah.
And I would think that EEG is more suited to capturing that unfolding of processes that happen in milliseconds, tens of milliseconds. Do you have any ideas about like experimental paradigms that we could use with EEG? I mean, of course, you're instantiating creativity in somebody without really knowing whether they're going to do it or not. Do you have some insight that you could offer on that?
I have two insights. So a lot of, and I neglected to mention this, but a lot of the early research in creative cognition was actually done with EEG. Razumnikova and people in Russia are great early people, Bektereva, people that have done EEG research before FMRI came even into existence, kind of in the early to mid-90s. So a lot of EEG research is available, and I would make a suggestion that MEG, magnetoencephalography, would be even better than EEG. It has better spatial resolution and all the temporal resolution, so you could transform, if you have a good fMRI experiment, you could transform this to a MEG environment and get all of the temporal resolution that you desire or all the spatial resolution that you desire with a massively increased temporal resolution.
With the understanding it's less portable, too.
Yes.
Rex, do you have intracranial recordings, given that you've got their heads open?
Yeah.
It seems like you're well positioned to have the best of all worlds.
Yeah, I'm not going to be Dr. Knight, but I think I'd like to start, and we're doing about one awake craniotomy a month now, and I'm the neuropsychologist, so I have an opportunity to actually do intracortical recordings, and we're going to start this process. I'd like to write a grant to do this process with one of the neurosurgeons who is very interested in doing awake craniotomies, so I'd like to report back to you about some success in the future.
Go ahead.
So,
Yes.
a question about the large study that integrated many different findings about creative normals versus less creative normals. So the quick question is, how did you measure creativity? The second is, some of the measures that you reported were structural.
Yes.
Brain structure. Some were process, right? How information processing online. And I wondered if you wanted to distinguish those.
So we used mostly measures of divergent thinking. So tell me as many different
How
ways
many
you-
uses of a brick?
And also the Creative Achievement Questionnaire was a second major technique that we used, and then measure of personality openness to experience. These are all measured outside of the scanner. We didn't do any measures inside the scanner. We have a 3-hour battery of behavioral tests that we administer, and then we did all structural imaging techniques in the scanner, and then we correlate them together.
But do you want to say something about the structure versus the processing? I mean, findings, creative solutions online?
I'm hoping that someone will do a good study that actually integrates the structure versus function. There's really not good literature that shows how this lower cortical thickness, for example, relates to functional techniques. I'm kind of waiting for a really bright person to actually do that study to see how they relate. Is lower cortical thickness, is that actually mean more cortical activation in an fMRI environment or less activation? Someone really needs to do those together in order to really nail that.
In the context of dementias, the relationship between structure and function has been well documented
Yes.
by Bill Seeley and colleagues, and that approach has been taken over and adopted by a number of investigators. So you can look at the correlations across individuals of brain structure at each voxel, and there's quite a nice relationship between those patterns and the patterns that emerge from the functional correlations. But again, that's certainly much more robust when there are very
Obvious.
severe processes
Obvious
that
psychopathology,
are in place. But that general scheme has been transported to other samples and other populations as well.
Yeah.
But as Rex said, it's also really a challenge to integrate these various measures. In part, they have different distortions. They're sampled in at different levels of resolution. So the structure is typically 1 millimeter cubed, and the fMRI is 3 millimeters cubed, et cetera. And so it's a non-trivial process. And where the fMRI signal can be ascribed to where it's coming from is still being discussed and investigated, for example. But let's take the final comment or question.
You talked about improvisation at different points. You had Miles Davis on one of your early slides and mentioned a sort of down regulation that goes on in that instance of jazz improvisation. And I was thinking, I guess perhaps maybe you could implicate the default mode network there, and it would make sense that it sort of echoes maybe things that artists have said or Charlie Parker has said, that you learn so much and then in performance, in the act of it, you forget it. I might play a line, but I'm not thinking 1, 2, flat 3, 4, 5, flat 3, 2, 1, 7, 5, 6. That's a common line, but I'm not thinking that.
Even in normal speech, you don't quite know where the sentence is going to end. You just know that you're going to begin it and it will end someplace. Now I'm thinking about my speech. You're going to begin it and it will end somewhere. Yeah. So it's an improvisational task just to speak.
But I was wondering, because you also mentioned like the 10,000 hours or 10 years that it takes, that information that you do build up and store is important to the way that you create. So I was wondering if those kind of years of experience and knowledge have any influence on the default mode network or whatever you might implicate. Does it expand or sophisticate it? Is there plasticity there?
That sounds like a fantastic study.
Thanks. And just one more in addition.
I'm not aware that it's been done.
Okay, well that's good. I'd like to do it.
Yeah.
But you had 4 boxes there, and I was wondering basically, would Madame Curie's sort of brand of creativity be antagonistic to Miles Davis's spontaneous and emotional brand? Because you said you can switch between them, but would them, an integrated version of those be workable or be antagonistic?
So it's my current bias, if you will, that there's this general creative, cognitive network or thing that our brain does that transcends, that is transdisciplinary, that it is perhaps slightly different depending on the discipline of an artist versus a jazz musician versus a scientist, but there's this central reasoning process or this central process of the brain that transcends those those particular disciplines. I could be disabused of that bias in the future as more data comes in, but that's my current working, well, we also call it a hypothesis.
Okay, thank you.
Yeah.
Let me bring this particular talk to a close by highlighting one of the early take-home messages that we would all be well served to really think about, which is that reliability limits our ability to obtain validity. And Rex's illustration of that was simple, clear, and forceful. And I think that really is the rate limiting, or the limiting factor in our work, is that we've inherited and we've adopted measures that are at best fair in their reliability, and that really is doing this with one eye firmly closed and the other one just barely open.